to start off the webinar by making a bold statement that individual molecules, mutations, or genes are insufficient as biomarkers for complex therapies like immunotherapy. A good example of an individual marker is PD-L1, the most commonly used diagnostic for PD-1 or PD-L1 checkpoint inhibitor therapies such as Pembro or Nivo. And in discussing PD-L1, the European Society for Molecular Oncology put it plainly that PD-L1 immunohistochemistry is not suitable as a biomarker for selection of anti-PD-1 or PD-L1 inhibitors. Moreover, the Society specifically recommends a multi-component predictive biomarker for patient selection of immunotherapy drugs. And current methods for characterizing the immune system attempt to use all possible biological materials and methods, but all of these only show modest ability to predict response to therapies. And the vast majority of clinical samples are stored as FFPE materials, which limits the technologies that are available for analysis. RNA sequencing specifically has shown value because it has the potential to tell you much more about the disease and its progression. This has led to significant investment in precision and personalized medicine, searching for an elusive signal in hopes of finding powerful biomarkers. But the truth is, there is no single RNA gene signal, fusion, or even pathway that can accurately predict a disease state. Diseases are far more complex than that. And this is especially true for cancer and immunotherapy. The inability of these technologies to comprehensively characterize immune response is likely because most are only looking at those individual markers, which are not robust. And as computational biologists, we at Cofactor recognize the power of big data and the complexity of biology. We've decided to apply the power of machine learning to RNA expression and develop a new multidimensional model for predicting cancer biomarkers. We invented a new discipline called predictive immunomodeling, which creates robust pictures of how to better predict a patient's response to therapies. We call these powerful representations health expression models. These health expression models go far beyond the scope of a single gene and include multiple facets of biology, looking both at the presence or absence of RNA, as well as the dynamic expression levels. These can be influenced by the state of the disease, environmental effects, therapies, etc. So this shift to a new way of modeling cells, patients or cancer, can be extended to, in the future, treatments, which can produce an accurate model of a patient's risk of cancer, their response to a treatment or drug, and the overall prognosis of their health outcomes. Specifically, in the ImmunoPrism assay, we've applied this approach to develop superior models for the immune cells which play an integral role in fighting disease, particularly in the tumor microenvironment. These immune cells have been shown to be powerful potential biomarkers for understanding response to immunotherapy, but methods to detect them have been semi-quantitative at best. We call each of these immune expression models by their more common names, such as CD4 positive T cell, Treg, etc., based on the original cell donors that were used to generate the models. So today, you'll hear about how these immune expression models are used in the ImmunoPrism assay to characterize the immune cells present in a solid tumor sample. The assay accepts all solid tumor materials and delivers a fully analyzed report, which quantitatively breaks down the immune cells present. And at the end of the talk, after you've learned about the assay and its performance, I'll share more about how this technology may be applied to biomarker discovery.